Hello everyone, welcome to our video presentation. Our topic today is Myosin. This presentation is prepared by 10 members.
My name is Siti Sarah Sufia, my check number A212355, and I will start by introducing myosin. So, what is myosin? Myosin is a major protein found abundantly in muscle tissue. It plays a very important role because it acts as a contractile protein, which means it is directly responsible for muscle movement. Inside our muscles, myosin is located within the myofibrils of muscle fibers. These myofibrils are made up of repeating units called sarcomeres, which you can see in the diagram on the right. During muscle contraction, myosin interacts closely with another protein called actin. Myosin heads attach to actin and pull it inward. This process is known as the sliding filament mechanism, where the filaments slide past each other, causing the muscle to shorten and contract. This movement does not happen for free. Myosin uses ATP as its main source of energy. ATP provides the energy needed for the myosin heads to attach, pull and detach repeatedly from actin. So, in short, myosin is essential because without myosin, muscle contraction and movement would not occur. Next, let's move on to the classification of myosin and its importance in food chemistry. Myosin is classified as a myofibrillar protein in muscle. It is also one of the main types of contractile proteins together with actin. Structurally, myosin is a high molecular weight protein with a complex shape, which allows it to perform its mechanical and structural functions. Besides movement, myosin also plays an important structural role in muscle fibers. It contributes to muscle function, strength and stability, making it essential for normal muscle performance. Now, looking at its importance in food chemistry, myosin is a key protein component in muscle-based foods, especially meat and meat products. It strongly influences meat texture and firmness, which affects how the meat feels when we chew it. During heating, myosin denatures and contributes to gel formation, helping processed meat products maintain their shape. It also helps bind and retain water, improving juiciness and reducing cooking loss. Because of these properties, myosin directly affects the quality, heat, and consistency of processed meats, making it very important in food processing and product development.
Hi everyone, my name is Fire Izwan A212277. I will explain about the animal sources of myosin. First one, we have the skeletal muscle. Skeletal muscle is the primary and richest sources of myosin. Common food animals such as beef, chicken, and fish contain high level of myosin because skeletal muscle makes up most of the edible portion of meat. These muscles are voluntary muscle and are heavily involved in movement, which explain the high myosin content. In food system, skeletal muscle myosin is the main functional protein contributing to texture and structure. Next one, we have the cardiac muscle, that is also called heart muscle, contain myosin but is less consumed as food. This myosin in cardiac muscle is more specialized for continuous contraction, although present, its contribution to overall food product is much lower compared to skeletal muscle. Next, we have the relative abundance of myosin in meat. Myosin is one of the most abundant protein in muscle, classified under myofibrillar protein. It accounts for a large proportion of the contractile protein system in meat. Compared to sarcoplasmic and connective tissue protein, myosin has greater functional importance in food processing. We have the relative abundance of myosin can vary based on the animal species like fish or beef, muscle type, active or less active muscle, and also can be classified by age and psychological condition of the animals. Next one, we have the relevance to food product. Myosin is highly important in meat processing because it is salt-soluble protein. When salt is added, myosin is extracted and form a protein network during the heating. This network is responsible for gel formation, improved water holding capacity, and emulsification of fat. For example, in surimi production, fish myosin is the key protein that form a strong elastic gel that give the surimi product their characteristic texture. Also, in processed meat like sausage and nugget, myosin help bind meat particle together, improving cohesiveness and mouthfeel.
My name is Nordina Salsabila binti Muhammad Bustaman and my matrix number is A212035. Today I will explain the primary structure of myosin. The first one is amino acid composition. The primary structure of myosin refers to the simplest level of protein structure. It is composed of amino acids that arrange in a specific linear sequence, forming a polypeptide chain. Different classes of myosin possess distinct amino acid sequence, which determine their chemical properties. The second one is molecular weight, where myosin is considered as a high molecular weight protein. This is due to its long amino acid sequences and the large number of amino acids that present in its structure. For example, class 2 myosin consists of two heavy chains with a molecular weight of approximately 220 kilodalton each and four light chains with approximately 20 kilodalton each. Next, the amino acids in myosin are linked together by peptide bonds. A peptide bond forms between the carboxyl group of one amino acid and the amino group of the next amino acid, creating a long polypeptide chains. Lastly, myosin contains heavy chains and light chains. The heavy chains consist of very long amino acid sequences and contribute to the majority of protein molecular weight. In myosin, two heavy chains call together to form a double helix. In contrast, the light chains have a shorter amino acid sequences and the function is to stabilize the head structure of the myosin molecule.
Hello everyone, my name is Zhang Junwen. My code number is A207978. My topic is secondary structure, alpha helix, dominance, and cold core motif. Let's talk about core factor first. Myosin's secondary structure is dominated by alpha helices, accounting for over 80% of the heavy chain structure. This forms the fundamental backbones of the protein. For cold core formation, two alpha helical heavy chains twins around each other in a left-handed cold core configuration, structuring the long roll-like tail region of myosin. Structural significance: the cold core is a key structure motif that enhances the protein's stability and elongated shape, which is essential for subsequent filament assembly. Next, total structure, goblet head, and stabilizing bond. The first point, domain distribution. The total structure consists of two functionally distinct domains. One, goblet head, a compact, spherical and terminal domain that contains the actin binding site and ATP as active center. These are critical for myosin's biological functions. 2. Roll-like tail. The C-terminal domain maintains the cold core alpha helix structure, ensuring structural rigidity. The second point is stabilizing bonds. 1. Hydrogen bonds. Fall between polar amino acid side chains and peptide backbones atoms, reinforcing the alpha helix structure. 2. Hydrophobic interactions drive non-polar amino acid residues to aggregate inward, reducing contact with aqueous environment and stabilizing the overfold.
Hello, my name is Zati Umar binti Usma. My metric number is A208987. Next, I will explain the quarterly structure of myosin. Myosin is a complex protein made up of several polypeptide chains that work together as one functional unit. It consists of 2 heavy chains and 4 light chains arranged in organized structure. The heavy chains form the main structure of the myosin molecules. They create the globular, head, neck, and long tail region. The head region is very important because it contains the actin binding site and ATPase site, which provide energy for muscle contraction. The light chains are attached to the neck region of the heavy chains. Their main function is to stabilize the structure and help regulate myosin activity during contraction. Together, the heavy chains and light chains form a stable, functional myosin molecule that is essential for muscle movement. Thank you. Now, I will move on to the conformation and filament formation of myosin. Overall, myosin has an elongated and fibrous shape which makes it suitable for forming muscle structure. During filament formation, the tail regions of multiple myosin molecules align and pack together. This arrangement leads to the formation of thick filament in muscle fibers. In terms of actin myosin interaction, the myosin head projects outward from the thick filament. These heads interact with actin filament during muscle contraction, allowing the sliding movement to occur. Functionally, the quaternary structure and proper filament formation of myosin are crucial for the sliding filament mechanism, which is the main process involved in muscle contraction. In conclusion, the structure of myosin is closely related to its function in muscle movement. Thank you.
Hi, my name is Wanzara binti Jamil. My metric number is 8208809. On this slide, I will explain the overview of muscle contraction. Muscle contraction mainly involves 2 proteins which are myosin and actin. Myosin forms the thick filaments while actin forms the thin filaments. These 2 filaments interact within a structure called the sarcomere. Contraction occurs when the filaments slide past each other, not because they shorten. As the filaments slide, the sarcomere shortens, which results in muscle contraction. This slide explains the first step of the sliding filament theory, which is when myosin is energized. Myosin has ATPase activity, which allows it to hydrolyze ATP into ADP and inorganic phosphate. ATP binds to the myosin head and is broken down into ADP and pi. This reaction releases energy, causing the myosin head to move into a cock or ready position. At this stage, the myosin head is energized and prepared to bind to actin. Next, the energized myosin head attached to the actin filament. This attachment is known as a cross-bridge formation. During this step, ADP and pi are still attached to the myosin head. This binding is important because it allows the next step of muscle contraction to occur. This slide shows the power stroke, which is the main force generating step. First, the inorganic phosphate is released, causing a shape change in the myosin head. Then ADP is released. This causes the myosin head to pull the actin filament towards the center of the sarcomere. As a result, the sarcomere shortens and the muscle contracts. After the power stroke, a new ATP molecule binds to the myosin head. This binding causes myosin to detach from actin, breaking the cross bridge. ATP is then hydrolyzed again, re-energizing the myosin head. The cycle repeats as long as ATP and calcium ions are available. This continuous cycle allows sustained muscle contraction.
Hi everyone, my name is Koja Xuan, metric number A208505. For this slide, I will talk about the role of myosin in muscle tension. Myosin is responsible for generating force in muscles. This occurs when the myosin head binds to actin filaments to form cross bridges. Using energy from ATP hydrolysis, the myosin head pulls the actin filament, which produces muscle tension. This tension is essential for maintaining muscle strength and posture. Moving on to the next slide, myosin also plays a crucial role in muscle movement. Through repeated attachment and detachment between myosin and actin, the muscle filaments slide past each other. This process is known as the sliding filament mechanism. As a result, muscles shorten and relax, allowing movements such as walking, breathing, and lifting objects. Lastly, myosin is not only important in muscle function, but also in other biological and food-related processes. In cells, myosin helps with intracellular transport by moving organelles. In food chemistry, especially meat science, myosin plays a significant role after slaughter. Changes in myosin structure during post-mortem processes affect meat texture, firmness, and water holding capacity, which directly influence meat quality. So, this shows that myosin is important both biologically and in food quality.
Hi everyone, my name is Wan Nur Musdalina binti Wan Mahmud. My metric number is A209399. So, I would like to talk about physical chemical properties of myosin. First is solubility of myosin. Myosin is a salt-soluble protein, also known as myofibrillar protein. This means myosin does not dissolve in pure water. In water, myosin molecules attract each other strongly and form aggregate, so they remain insoluble. When salt such as NaCl is added, the salt ion reduces the attraction between myosin molecules. This allows the myosin filament to separate and dissolve in the solution. Usually, myosin becomes soluble at moderate to high concentration, around 0.3 to 0.6 molar NaCl. This property is very important in food system, especially meat product, because soluble myosin helps in water holding capacity and gel formation, which improve the texture. Second is effect of ionic strength. Ionic strength refers to the concentration of ions such as sodium and chloride ion in a solution. At low ionic strength, there are fewer ions present. As a result, protein-protein interactions are strong, causing myosin molecule to aggregate. This leads to low solubility of myosin. When the ionic strength increases, more salt ions are present in the solution. This ion neutralize the electrostatic charge on the myosin molecule. This reduces attraction between the protein, allowing myosin filament to dissociate, which increases solubility. This property is very important in food processing, especially in products such as sauces, surimi, and restructured meat product, where soluble myosin contributes to good texture and binding. However, excessive salt can negatively affect flavor and may cause excessive protein unfolding, which reduces product quality. Therefore, increasing salt concentration increases myosin solubility only up to an optimal level. Third are pH sensitivity and isoelectric point, or PL. Myosin is sensitive to changes in pH. pH affects the net charge on myosin molecules and also influences protein-protein interaction. When the pH becomes very acidic or very alkaline, myosin can undergo protein denaturation. This denaturation reduces its functional properties such as solubility and gel forming ability. The isoelectric point, or PL, of myosin is approximately pH 5.4. At this pH, the net charge of myosin is 0 because there is no charge. Protein-protein attraction is highest, causing myosin molecules to come closer together. As a result, myosin has the lowest solubility at its isoelectric point. When the pH moves away from the PL, either higher or lower, myosin molecules carry a net charge. This increases electrostatic repulsion, leading to higher solubility. Therefore, myosin is least soluble at pH 5.4, which is its isoelectric point.
Hello, my name is Nurul Suhaila binti Azmi and my metric number is A211366. After discussing the basic physical properties of myosin, I will now focus on the advanced The other physical properties, specifically the thermal denaturation, gelation, water holding capacity, and emulsifying ability, which are highly important in the food processing and meat product quality. Myosin is thermally sensitive muscle protein that undergoes denaturation when exposed to heat. So thermal denaturation usually occurs between 40 to 60 degrees Celsius, where the alpha helical structure begins to unfold. During this process, hydrogen bonds and hydrophobic interaction that stabilize with the protein structure are disrupted. So as the result, myosin lost its native conformation and exposed to reactive groups, which allow the protein-protein interaction to occur. So this thermal denaturation is crucial in the food chemistry because it initiates the structure change that later contribute to the texture development in the cooked meat product. Following the thermal denaturation, myosin exhibits the strong gelation properties. When heated in the presence of the salt, denatured myosin molecule aggregate and form a three-dimensional protein gel network. So this gel network is responsible for the firmness, elasticity, and cohesiveness of the processed meat product. So the strength of the gel form depends on the factors such as cell concentration, heat temperature, and the protein content. Therefore, myosin plays as a key role in the gel formation in the products such as sausage, meatball, and surimi-based food. In addition to gelation, myosin contributes significantly to water holding capacity. The gel matrix formed by myosin is able to trap the bind water molecule, which is reduce the cooking loss and improve the juiciness and product yield. Myosin also functions as an effective emulsifying agent because it contains both hydrophilic and hydrophobic agent. These properties allow the myosin to stabilize the fat-water immersion, which is essential in the emulsifier meat products such as frankfurters, nugget, and sausage. Water holding capacity is also affected by pH. At the isoelectric point, the net charge of the myosin is minimal, which reduces the water binding and result in the lower water holding capacity. Conclusion: the physical properties of myosin, particularly thermal denaturation, gelation, water holding capacity, and emulsifying ability, are critical determinants of the texture, juiciness, and stability in the processed meat product. These properties highlight the importance of myosin as a functional protein in the food chemistry and the meat science.
Hi everyone, my name is Myosin Mariam and my metric number is A210333. So today I'm going to talk about a protein that absolutely essential in food science, especially when it comes to the texture of the meat and seafood products we eat. That protein is called myosin. In living muscle, myosin job is to enable contraction, but after the slaughter, it plays a very different role, where in the process called rigor mortis, myosin form a very tight bond with another protein called actin, which make the meat initially very tough. However, if we allow the meat to age, enzymes slowly break down this protein, including myosin, to improve the tenderness. So the state of myosin filaments is a key factor in the initial toughness and eventual texture of the fresh meat. Next, myosin's most important functional property is something else, which is its ability to form a heat-induced gel. And here how it works. Okay, so when myosin is extracted, often using salt, and then are heated, its structure will be unfolded. And then when it cooled down, it will reform into a continuous three-dimensional network. And this network is actually pretty strong, elastic, and it traps water incredibly very well. For example, you can think about the firm, bouncy, sliceable texture of a sausage or a ham. That's the myosin gel holding the meat pieces together. Or you can also consider surimi product like imitation of crab stick. And now we're talking about the effect of heating and the effect of salt and ACI. The quality of this gel depends on two key factors, which is heat and salt. And for heat, optimal gel strength happens between 50 degrees Celsius to 70 degrees Celsius. And too much heat will cause synersis where the water will squeeze out, leaving a dry, rubbery product. And that's why slow cooking sausage to an internal temperature around 70 degrees Celsius is pretty important. For salt, salt does two things. First, it attracts myosin from muscle fibers and creating a sticky paste that coats meat particles. Second, it increases ionic strength, helping myosin to dissolve and form a very stronger, more elastic gels during heating. To wrap up, Myosin is much more than a biological motor protein. Its unique structure allows it to dissolve in salt and form a heat set cells with excellent water holding ability. And this property also makes it indispensable in food manufacturing, responsible for the binding sausages, the sliceability in ham, and the bounce in surimi. By controlling salt and heat, food scientists can also design the texture that range from tender to firm, meeting consumer expectation in product worldwide.